So this will be on chromosomes and chromatin. This is our week 7 lecture. So let's begin. Before we get started, we'll just have a quick recap of what DNA is. So remember, DNA it's a double helix. So we have 2 complementary strands that has a pentose sugar backbone. And it's made up of 4 nucleotides, which are adenine, thymine, guanine, and cytosine. And they complementarily pair with one another. So adenine always goes with thymine, and guanine will always pair with cytosine. And remember, DNA, this is the blueprint for our life, for all life forms. Okay, so you can go down from a prokaryote going all the way up to eukaryotes. This is the genetic information of all organisms. And the other thing about it is, it has the ability to replicate through transcription. So it can self-replicate. It can make copies of itself as well. So here's our double helix. So we got one helix going over here, and another helice going over in this direction. Remember, they are anti-parallel. So one goes from this one, for example, goes from a 3' to a 5' direction. The other is going to go from a 3' to a 5' direction. So they're anti-parallel. The strands are anti-parallel. So the other thing that we see over here are these areas over here. So we got one big wide, if you kind of imagine this as like an arrow that's pointing in, we have a big arrow. Then we also have another one on this side that's little. So we have major grooves, which is what this one would be, the big one would be, the larger one. And then we have smaller ones, the minor groove. So there's a minor groove here, a major groove over here. There's another minor groove over here. And again, on this side, we have minor and major grooves. So as for the base pair, so remember, we have the sugar phosphate backbone. And you can see these are our nucleotides over here. So we have, for example, this is guanine, and this would be cytosine. So anytime you see guanine, there's cytosine. So there's cytosine here, guanine there. And the other would be adenine and thymine. So here's thymine over here, and here we have adenine. So again, you'll see these occurring over and over again. Again, we have an adenine here, thymine over there. Same thing over here. You have the adenine here and the thymine over there. So again, these are the complementary base pairing that we talked about. And over here you can also see, again, major and the minor grooves. So when we think about DNA, everybody thinks about this X shape, this chromosome structure. Now keep in mind, the only time we see this structure over here is when the cell is dividing. Okay? So when the cell wants to divide, it wants to make a copy of itself, this is when this structure will appear. Other times you do not see this structure. Okay, this genetic information, it's not super condensed into this form, to this X shape that we see all represented most of the times. Now, with that said, let's just look at this supercoiled, this structure over here, the super condensed structure, and we have different parts. So the parts on top, okay, these, if you kind of look at it, one thing to remember is this. This is actually made up of 2 chromosomes. So we have one chromosome over here and another one over here. So in other words, 2 chromatids. So we have a chromatid this way, another chromatid over here. And they get joined together. They're combined by this region over here, which is the centromere. And the ends of this are called the telomeres, right? So we have telomeres at the end. These are the regions at the end, telomeres. So one chromosome has 2 telomeres. We've got 2 over here and 2 over here. We've got a centromere over here in the middle. And we have arms that you see over here. So if you look over here, this arm is shorter. This one over here is much longer over here. So the short arm, we call it the P arm, and the long ones are the Q arms. Okay? So remember, we have 2 sister chromatids that make up a chromosome, which are held together by a centromere. And this chromosome has the short arms, which are the P arms, and the long arms, which are the Q arms. Another thing that we can say about chromosomes are the different types based on where the centromeres are located. So in this drawing over here, you can see the centromere is all the way at the end of a pole. And so we call this a telocentric arrangement. Okay, so this is a telocentric chromosome. Then when you come over here, you can see that the centromere is located close to the pole, but not all the way at the end of the pole, as we see in the telocentric. So we term this to be acrocentric. Other times, we see the centromere to be not so close, not all the way at the end of the telomere, and not even close to the pole, but somewhere in between the middle. So it's like slightly off the middle. So we call that a submetacentric. Compare that to when it's directly in the middle, then we would call that a metacentric arrangement. Okay, so again, once again, you can see the short arm and the long arms over here. So you can see over here in a submetacentric, the short arms are much shorter than in the metacentric. And then when you come to the acrocentric, we have very, very short P arms, and the Q arms are rather long. And then in telocentric, there's an absence of the P arm, and where we just see Q arms that are shown. Chromatin can be of two types. We can either have euchromatin or we can have heterochromatin. So as for euchromatin, euchromatin is lightly packed, okay? And whereas heterochromatin is much more condensed, okay? So it's a lot more condensed, meaning it's much more tightly packed. So again, euchromatin is lightly packed, it's less condensed, and this is active. Okay, the gene is going to be active over here. So we have it's active in gene translation, and also it's gene rich. It has a high GC content that we see in euchromatin. And also when we stain it, it ends up staining much lighter. Okay, so we have it stains lighter. When we compare that to heterochromatin, again, as I said, this is it's more condensed, and it's whereas the euchromatin was active. In other words, it was active in gene translation, meaning this holds genetic information. Okay, so this is where a lot of the information is coming to make the proteins and whatnot. All the orders are coming from over here. So again, this is very rich in information. So it's gene rich. Again, this is why I said it's high in GC content. Heterochromatin, again, more condensed, and this is going to be non-coding. Okay, heterochromatin is non-coding. And so again, it's gene poor, so it's going to be high in AT content, and comparatively, it's going to stain darker. Okay, heterochromatin will stain darker compared to euchromatin, which stained lighter. And heterochromatin is going to be, as I said, it's much more condensed. And we're going to see that there's actually two types that we're going to be looking at. Two types of heterochromatin. And we'll talk about that in the next slide. So when we look at this photograph over here, so this is essentially the nucleus, and this is all the genetic information inside. This is all the DNA or the chromatin. So what do we see are these light areas, and then you see the dark areas. So remember what we said before. As far as euchromatin goes, euchromatin is lightly packed, okay, and also that it stains light, okay. It doesn't stain dark. So when you look over here, all of this is all euchromatin, okay, and this is going to be rich in it's going to be high in GC content. These are active genes, okay. So there's translation that's going to be taking place. Then all that stuff, so remember, this is that nuclear membrane over here, this part all around, that's the nuclear membrane. And next to the nuclear membrane, and then also we have certain areas over here, where we see dark staining. This is all heterochromatin. So remember, this is non-coding. It's gene poor. It's high in AT. So it's much, much, much more condensed. And that, you can see again, it's next to the nuclear membranes. Look found throughout the nuclear membrane. Then also we see it in certain parts of the nucleus as well. Okay. All right. So a couple of slides ago, I told you that heterochromatin is of two types. So of those two types, our heterochromatin could be either constitutive or it could be facultative. So for the first type, over here, there is some control that's exerted. So remember what I said, heterochromatin, initially, first I said that, it's non-coding. It doesn't code for anything. True. However, it may or may not be completely silent. So in the event of here, let's just start off over here. For facultative, it's completely silenced, okay, by histone deacetylation. Nothing is happening over here, all right? Compared to the constitutive heterochromatin, we find this at the centromere, we find them at the telomere, and what we find is that they tend to, for the gene sequence that's besides it has some influence over it. So it does control, it may be able to control some aspects of it. So in that sense, again, constitutive heterochromatin, it's not completely silenced as to facultative, which is completely silenced because of the histone deacetylation. All right, so why does DNA need to be packed? So remember when we went a handful of slides ago and looked at this, or actually even over here, this is all the genetic information, right? And this is actually, this is somewhat condensed. Remember what I said? I said this is tightly packed and this is loosely packed. Now, why do we need to do that? Well, if you look over here, again, what is this? This is the nucleus, all right? And if you look at it, this is already full. Now, what is the size of this area over here? What is the size of the nucleus? The nucleus is about 5 to 6 micrometers. That's tiny. So to kind of give you an analogy of how small the nucleus is, think about if you were to take a marble and you would put that marble in a football stadium. That marble would be the nucleus. All right? Now, so again, 5 to 6 micrometers is very small. What do we have inside there? Our entire genome needs to fit inside there. And DNA, it's not small, it's big. If you were to stretch it out, we're talking about 2 meters long. So how do we fit something that's 2 meters long? Again, something that consists of 23 pairs of chromosomes. How do we condense something that's 6 feet long inside an area with a diameter of 5 to 6 micrometers? How do we fit 23 pairs of chromosomes inside there? Well, we have to pack things up. And this is essentially what this unit is about. How does that happen? And again, we kind of explain as to why that needs to happen. But how do we do that? So that is what we're going to be talking about for the remainder of this unit. So when we're talking about packaging this genetic information, how does that take place? So again, we start off with our genetic information, our DNA, and what ends up happening is we see that DNA, it starts to wind up into loops around these structures, which we're going to be looking at. So these structures, when they wrap around them, we get a different term. We now start to call them a nucleosome. So nucleosome essentially is DNA wrapped around this bead. Let's just call it a bead for this time. Now, as we progress, what ends up happening is these beads, they start arranging themselves in different orientations, in different thicknesses. And once that happens, again, we have a different term for it. We call that a chromatin, a fold over here, or chromatin threads we start to see. Then what we start to see is that these 30 nanometer chromatin fibers, they start to loop up. Okay, they start to arrange in a different structure. Okay, so now we go from them looking like beads on a string, and then they start to have these 30 nanometer fibers that they start to appear in at that point. Then they start to form these loops, these 300 nanometer loops that they occur in. And then what ends up happening is these loops, they start to condense even further. Okay, so they start to get wider. And then from that point on, again, they start to coil up even more. So they start to supercoil. And that's essentially what we end up having over here. At this point, they start to compress and arrange themselves on these scaffold proteins, which eventually leads us to this chromosome structure that we see. So we go from here where, again, you have DNA, all right, just DNA by itself, and then we start to progress. It starts to get tighter and starts to get even more tighter, and after that point it starts to rearrange and starts to compress further and further until it ends up taking its final shape in its final compressed form, this supercondensed form, into what we see as chromosomes. Okay, so let's talk about chromosome packing. Now, what exactly is chromatin? Let's define that. And earlier on I told you it's just DNA and some protein. So again, DNA plus protein equals chromatin. Okay, so DNA and protein is chromatin. What exactly is this this protein? The proteins that we're talking about are histones. So when you take genetic information, DNA, and they get wrapped around these histone proteins, we call them chromatin. And again, they appear to look like beads on a string. So again, histones are proteins that are important in maintaining the chromatin structure. When we talk about a nucleosome, so this nucleosome is just a model. It's just a model of the model of a wrap as it's around the DNA around an octomer. We'll be looking at photographs of that also. And remember what I said, chromatin, it's in a semi-condensed state. When you look at the chromosome is said to be in a supercondensed or a supercoiled state. And again, we only see this when the cell is dividing, okay? So the chromosomes are only seen when cells divide. That's when they're in this supercondensed, supercoiled state. The rest of the time, the genetic information, it's seen in a semi-condensed state in the form of chromatin. Okay, so chromatin is semi-condensed. It's wrapped up, it's packaged, but it's not completely unpackaged. Okay, it's not completely loose. So we have, again, why is that possible? Because there's just so much of that information that needs to, remember, we have 2 meters worth of DNA that needs to fit inside 5 to 6 micrometers. So we need to have it packaged up. So DNA, genetic information, for most of the time, it's going to be in a semi-condensed state. So as far as histones go, remember, histones are proteins. So let's just go back a couple of here. There we go. This is what we're talking about. This is a histone. Okay, this is a histone. This is a histone octomer that we're looking at, and we're going to be talking about what that is and what that means. But histones, they're found in eukaryotes. Okay, they're highly alkaline proteins that package and order the DNA into structural units called nucleosomes. Okay, so here we go. This is a nucleosome over here. When we have an octomer and the DNA is wrapped around it, this is the term that we use. A nucleosome consists of 8 histone proteins. So again, histones, this is the first unit that helps condense that genetic information. So once again, remember, these are basic proteins that we find associated with DNA and help the DNA to condense into chromatin. So when you look over here, some of these histones, they function as a spool for the thread. Imagine this as being the thread, for the thread-like DNA. Okay, so some of these histones, they function like a spool for them to wrap around. And you can see them again. This is wrapping around. The thread is the DNA, and the DNA wraps around this spool. And what is this spool? The spool is the histone. And if you were to count the number of proteins, we're going to be talking about these histone proteins a little bit, but we have about 8 of these over here. So 1, 2, 3, 4, 5, 6, 7, 8. This is why it's called an octomer. So again, when you look at this over here, so when you look at this chromatin and you find it wrapped around, and notice there's 2 wrappings. It loops around this twice. We call this a nucleosome. Okay, so this genetic information wrapped around twice, 2 times around this octomer, this histone octomer, forms a nucleosome. So we're going to be talking about the different parts of this. We might be coming back to this slide in a little bit. But remember, when we look at this again, when they talk about beads on a string, this is what we're talking about. Nucleosomes appear to look like beads. These are the beads that are on this DNA string. All right, so now that we have an understanding that DNA wraps around these histones, so these are the basic structural units for condensation to occur, for the genetic information to get packaged. Let's start talking about these structures and what they're made up of and what they are. So again, these histones, they're proteins. They're globular proteins that are made in the liver, all right? And they're rich in arginine and lysine. And we have, so aside from that, they have N-terminal tails, which plays a role in signaling and control of gene regulation. We have 2 major classes of histone proteins, all right? We have core histones, which are what these are, core histones, and we have linker histones, okay? Core and linker histones. There's 5 main types of them. So of the first group, the core histones, okay? Of the first group, the core histone proteins, there's 4 of them, okay? Those are H2A, H2B, so if you look over here, H2A, H2B, H3, and H4, okay? H2A, H2B, H3, and H4. These are the 4 core histone proteins. The other one, the 5th type, other one, are linker proteins, or linker histones. So the linker histone is H1, and we don't have a picture over here, but I believe we're going to be looking at it later on. The other thing to understand is when you look over here, again, as I explained earlier, this is made up of 8 histone proteins, okay, to form this, to form an octomer. So again, if you look at it, 1, 2, 3, 4, 5, 6, 7, 8. And we'll be looking at this in a little bit more detail as we progress through the lecture, how this occurs and why this occurs, what its conformation is. So in terms of their numbers, chromatin contains roughly an equal number of H2A, H2B, H3, and H4, and about half the number of H1 molecules. Chromatin also contains diverse groups of non-histone proteins that play a variety of enzymatic, structural, and regulatory roles. So, again, as I was trying to explain to you earlier, two of each of these core histone proteins assemble to form one octomeric nucleosome core. Okay? And wrapped around this octomeric, okay, so remember now, once we get the DNA wrapping around it, wrapping around this octomer, we end up calling that a nucleosome. Okay? So, what we find is that the length of genetic information that's wrapped around is about 146, 147 base pairs. Okay, so you have 147 base pairs of DNA that's wrapped around. The part that's not wrapped around is called the linker DNA. So roughly from the start to this part over here, we're looking at perhaps about 200 base pairs that we have. But again, this is about 146, 147 base pairs long. In this slide over here, this is just a table for the histones that we spoke about and some of their properties. So remember what I said, this first one, this is a linker histone, whereas these four are the core histones over here. Okay, so H2A, H2B, H3, and H4 are the core histones. In other words, they form the core which the DNA wraps around. Okay, so these are the core histones. The linker histone, linker protein, is going to be the H1. And then you can see H1 is the heaviest. Okay, 21, when you look at the molecular weight, it's the highest out of all these. How large is it? Well, again, H1 tends to be larger, much larger than the rest of them. It's made up of about 223 amino acid residues. Again, you can see that actually all these are very rich in lysine and arginine. So again, you look at H2A, its molecular weight is roughly the same as H2B. And even in terms of amino acids, there's not much of a difference, but 4 amino acids difference between the two. H3 and H4 on the other hand, again, H3 is slightly larger than H4. Again, it consists of 135 amino acids compared to 102. And H3 is also very rich in lysine compared to H4, where they both share nearly an equal amount of arginine. So over here, what we're looking at, again, we're looking at the blue is the genetic information, the DNA, and these are the discs. And again, the discs represent the octomer, the histone octomer over here. Now, the other thing that you see over here is you see these A's and P's and M's. So the A's are for the acetyl groups, the P's for the phosphoryl groups, and then the M's that you see over here are the methyl groups. And these are important in how they interact with other proteins. So to kind of explain this to you, this is important in histone remodeling. So for example, again, now what do we have over here? Remember, the reason we did this, the DNA is wrapped around these cores, is to condense them, to compact them, right? To make it nice and tight, so everything can fit inside the nucleus easily. But what about when we need to access this genetic information? When we need to copy something? Or what if there's some type of a mistake, something's wrong, something that needs to be fixed over here? How do we fix it, or how could you read this information if it's so tightly bound, if it's compressed? Well, this is where these guys come into play at that point. Other proteins can come and start to interact with them, which essentially will change the conformation. In other words, it will change how tightly they're attached, or in some cases, they will completely dislodge from the chromatin, from the DNA. All right. So sometimes, when you get with specific interactions, the entire disc can be removed, and then you have this DNA that's all loose, and at that point it could get fixed or it could get read. Other times you don't have to completely eject this octamer. You just need to loosen it up, and that happens as well. That can do that as well. So under different situations, different proteins will come. They'll start to interact with these groups to bring about a change in the conformation, how they attach themselves to the genetic information, to the DNA. So yes, once again, when you're looking over here, this photograph versus that photograph, remember what we said in terms of euchromatin and heterochromatin? So this, remember, this is it has a lot of information over here. So this is, it's high in transcription. Okay, so transcriptional activity it's going to be high. So therefore, you can't condense it that much. So it's loosely packed. So when the time comes, you need to get some information. You can easily you can make this even looser, for example, and then start to translate as you need to go about. Whereas this part over here, this part doesn't do anything. So why should there be so much room over here? Why should it be loosely packed when we're not really going to, we need this stuff, but we don't need to use it all the time, or they don't have that much of a function in terms of transcription. Therefore, we can kind of pack it up and kind of put it away for good, so to say. So they end up getting packaged much more tightly. So they're much more densely packed. So if you look at, remember the linker DNA, we have quite a bit of linker DNA between these two nucleosomes. Compared to over here, it's very short. The other thing that you see in heterochromatin is that heterochromatin, the histones are highly methylated compared to euchromatin where they're not. So again, this is another difference between euchromatin and heterochromatin, and also what some of the properties are for these histones, okay, in terms of the groups that they have, acetyl groups, phosphoryl groups, and the methyl groups, and the role that they play in remodeling for the histones as well. All right, so let's start off by talking about H2A. So H2A is important for packaging DNA into chromatin. All right. Another thing is that so since H2A packages DNA molecules into chromatin, the packaging process affects how the packaging process occurs will affect the gene expression. Okay, so H2A has been correlated with DNA modification, right? And it also plays a major role in determining the overall structure of chromatin. And it's also been found to regulate gene expression. Okay, so that's H2A histone. Now, as for the next one, for H2B, H2B is also involved with the structure of the nucleosome of the beads on a string structure. Okay, so in addition to that, it's also an important protein in the field of epigenetics, and is thought to play a role in the dynamic and long-term regulation of genes. The H4 histone is also involved in the structure of chromatin in eukaryotic cells. So again, it's a structural component of the nucleosome, and it's subject to covalent modifications that include acetylation and methylation, which may alter the expression of genes located on the DNA that's associated with the parent histone octamer. H3 is also involved in the structure of chromatin. It has a main globular domain, and it has a long N-terminal tail. Now, histone proteins are highly post-translationally modified. However, when it comes to histone H3 is the most extensively modified of the 5 histones. Aside from that, it's also an important protein in the emerging field of epigenetics. And so its sequence variants and variable modifications they're thought to play a role in the dynamic and long-term regulation of genes. All right, so remember, we had core histones and linker histones. So far, we've only described the core histones, H2A, H2B, H3, and H4. Those are all core histones. Now we're going to be talking about the linker histones, and the most prevalent of them is H1. So H1 is one of the 5 main histone proteins, again, which are components of chromatin in eukaryotic cells. Though they're highly conserved, nevertheless, they're the most variable histone in sequence across species. They do not make up the nucleosome bead. Instead, it sits on top of the structure that's keeping in place the DNA that's wrapped around the nucleosome. Okay? And as I said earlier, they're present in half the amount of the other 4 histones. Now, as for H5, these are individual proteins that are involved in packaging of specific regions of DNA. That's H5. Kind of back up a little bit and go over a little bit. Now, again, we take genetic information and proteins. We call that chromatin. And again, remember what we said. When we have this chromatin, when they're wrapped around this core of 8 histone proteins, we call that a nucleosome. And these nucleosomes, they get packed to form these chromatin fibers. Okay, and these fibers, they start to take different shape. They start to condense into another type of a structure, and that structure further condenses to form another type of a structure, which then eventually compacts into this chromosome, this metaphase chromosome. All right, so when we look at the levels of packaging, essentially we have 4 levels of packaging. So that first level, again, is the nucleosome. And again, what is a nucleosome? We start off with DNA and wrap it around 2 times around this histone octomer. And that's what this nucleosome is. And again, they look like beads on a string. So the next thing that happens is, these beads on a string, they start to kind of fold up on themselves. They form this structure which looks like a solenoid. So this is this solenoid, this chromatin level of folding. So this is like the second level that we have of folding. Okay. Then once we take that, now we go from this more compacted, further condensed state, okay, the solenoid state, into the third level of packaging. And this third level of packaging, we start to have loops that form. Okay? And so these chromatin loops are the third level of organization, or the third level of condensation, the loop domain that we have. After that, again, these loop domains, they start to scaffold on, they start to attach themselves to these scaffolding proteins, and then they condense, they super condense into these tighter structures, which again we end up seeing in the metaphase chromosome over here at this point. So again, these are the 4 levels of packing that we have. This is the most condensed, the chromosome, and the least of them, and again, where it's transcriptionally active, would be in the form of the nucleosome. And remember, so again, if you kind of talk about heterochromatin and euchromatin, remember what we said? Light versus dark staining. So if you kind of look at this area over here, this is very dark, that's at the centromere. And again, centromere is mostly heterochromatin found over here. Compare that to the lighter areas, we have euchromatin that's located over there. So if we use a little bit of the knowledge that we have now about histones and these the histone octomers that are formed, and let's try to take that and apply that to euchromatin versus heterochromatin. So again, if you look over here, this is the part that's being transcribed. This is the active part of DNA, of the gene. So when you look over here in euchromatin, notice that again, you have the nucleosomes that form. However, it's more relaxed. So there's space, there's room, you can use this. Compared to the heterochromatin, notice how condensed they are. They're very condensed. Okay, and to the point where, again, this is one of the reasons why they say it's been silenced. Okay, so when it's super condensed in this form, you can't work with this information. So again, another way to look at it, again, when you need to have information, when you need to work with information, you cannot have it in such a compact form. It needs to be more in a condensed form. Okay, it needs to be in this active form. And again, the euchromatin is that active form. And then again, another reason why when you look at chromosomes, the only thing that's the reason that you turn into this shape is for a division to take place. Okay, so remember, what's going to end up happening in anaphase? Anaphase, you end up having these sister chromatids that start moving to the opposite poles. So the main reason you ended up condensing and coiling this DNA into the structure is for division. There's no work being done over here. So at this point, all this chromosome, when it's in this state, when it's condensed, when it's super condensed, it's not working anymore. It's just there. It's that all this information has been packaged just for transfer purposes. All right. So again, euchromatin versus heterochromatin. Heterochromatin information, it's not coding for anything. Okay, so it's silenced, whereas euchromatin, it's active. Okay, it can transcription can take place over here. So one other thing that I want to show over here is that in euchromatin, when you look over here, notice that you have these. So again, you have two loops. However, again, this is still condensed. Now, in order to work on this area of this DNA, it's going to be very difficult if it's tightly bound to this. So you need to be able to access this. So we're going to be talking next about how some of that access takes place. How can we loosen this up a little bit so we can get to this? We can copy we can actually copy this information. Or what if this protein goes bad? What if this histone, something goes wrong with that histone protein? What do we do in that case? So we're going to be looking at that as well in the next slide or the next couple of slides later. Okay, so let's talk a little bit about some of the things that we went over. Now remember, we're talking about taking DNA, which is 2 meters in length, right, into an area that's 6 micrometers, right? So again, the analogy that I gave you was that you're looking at maybe a marble being the nucleus, and it's inside this cell, all right? And if you think about the cell could be like, think about the cell as a football stadium. So if you were to take, that tiny little marble, and you think about fitting in 2 meters worth of material inside, how does that get? How do we achieve that? And we said, this gets done by folding up that genetic information, right? Compacting and condensing it. So how does that condensing of that information take place? We said that we have these proteins called the histone proteins that end up playing a role in that. They facilitate it, they allow for this to take place. And we said that there's 5 histone proteins, which are H2A, we have H2B, And we said we have H3 and H4. Okay, so we have these proteins. Now, and what did we say? We said that the DNA, it will come in and it's going to form loops around these proteins. So imagine this shaded area that's in blue, those are the histones, and the black, that's the DNA. Okay, that's wrapped around it. So this is where these guys play. These are the blue things. Now, how do they form? How do they combine with what? How do they interact? This is what I want to explain over here. So essentially what we end up having is that we end up getting dimers that form. So first of all, an H2A and an H2B protein, they come together and they connect with one another. Okay? So we have an H2A, H2B dimer that forms. So we call this H2A, H2B dimer. Okay? Now, we have another H2A, H2B that come and also form an H2A, H2B dimer. Okay, so now we have one dimer here and one dimer here. Now these two dimers form what's called a tetramere. Okay, it will form a tetramere. So now we have an H2A, H2B tetramer. Okay, so remember we have 4 different types of histone proteins, right? So far we just went over these. We get the same thing happening with H3 and H4. So for H3 and H4, we have H3, H4, they form a dimer, and then we have 2 more, H3 and H4, they also form a dimer. Here, let's go back over here. So we got a dimer over here. So now these 2 dimers, they end up also forming a H3, H4 tetramer. Right, so this dimer and this dimer will link up to form a tetramer. Just the same as these guys did. This dimer here, this H2HD, these 2 H2A, H2B dimers, they linked up to form a tetramer, and we have another one going on over here with H3 and H4 this time. So now we have one H3, H4 tetramer, and we have one H2A, H2B tetramer. Okay, so what ends up happening is this tetramer and this tetramer, they combine. Okay, now they will combine to form an octomer. Okay, oct means 8. So again, what do we have? If you count it up, 1. Let me change this out to here. Let's go. 1, 2, 3, 4. We have 5, 6, 7, 8 histones in total, right? So the sum of these 8 that are now combined, that are working together, form this octomer. All right, so this is where we end up getting this histone octamer that forms. Okay. So let's take a look at the histone octamers now. So what I was drawing in the last slide, I told you that when you have an H2A and an H2B histone come together, they form a dimer. So here's one dimer. Behind that, we have another dimer. We have another H2A and an H2B. So we have a dimer in the back and a dimer in the front. So when these two dimers come together, they form a tetramer. Then we come and look on top. We have an H3 and H4 dimer, and behind that we have another H3 and an H4 dimer. So again, we have these two dimers that also form a tetramer. Now we got a tetramer on top and a tetramer on the bottom. When they come together, they combine, they form this histone octamer. This is that bead which the string wraps itself around. Okay, so beads on a string. Now what holds these beads on the string are these guys here. This is the H1 histone. So if you look at this picture over here, here's the string, or in other words the DNA. DNA is wrapping itself around the histone octamer. And now what's going to hold it in place? What's going to stabilize it? What's going to make sure that there's enough tension between the first fold to the next? One of those things is this H1 histone. Okay, so that's what it's doing over here. It's keeping this in place, in position. So again, remember, the beads are the histone octamers, and they're made up of two tetramers. Okay, so it's a complex of two tetramers, or four dimers, if you want to look at it another way. What do we have around? We have DNA that wraps around it. DNA, again, that's that string. So roughly 146 base pairs wrap around it. And this part over here, this is the part that doesn't get wrapped around. This is the linker DNA. Okay, so it's going to link this nucleosome to this nucleosome. So remember what we said. When you have the strings in the bead, that's the nucleosome, right? So this is what we're looking at. So again, when you look at this photograph over here, essentially DNA is wrapping itself around twice from histone to histone to histone. So hopefully this makes sense of how this condensation is occurring, how the genetic information is compacting. So this happens in an orderly manner. You know, it's not just random, but you know, again, there's a system behind this also, and there's a system behind the folding, the condensation, the coiling that further takes place as well. So hopefully this is all starting to come together now in terms of the role of the octamers in condensing this genetic information, in compacting that in the DNA. So let's move into the final area of topic in terms of histone modification, histone remodeling. So again, if you look over here, again, what do we have here? We have genetic information, the DNA that's wrapping around this histones. And again, this is in a condensed state. It's a lightly condensed state. So what does that mean? That means that the genetic information is still packaged, still wrapped up. And again, when things are packaged, they're hard to access. You can't really do much with it, right? If you think about, you know, for example, if you're packing your suitcase, you're ready to go on vacation, you got all your shirts and on the bottom, and perhaps your jeans on top of that, and maybe you put some socks on top of that, and on top of that you put your swimsuits, and then your underwears. If you have it all compacted like that, you have it packaged and it's inside that suitcase, but you need to get all the way to the bottom where your shirt was. Well, you can't just really slide your hand in there and take it out, because it's everything's on top of everything. It's compacted, so you have to kind of shift things around a little bit, and then kind of lift up the top part, and then slide out the part that whatever you need. So similarly, in that sense, think of it like, the DNA is wrapped around. You need to attach some other DNA machinery. For example, if there's a break over here, or again, you need to, some transcription needs to take place at this region. How do we do that when it's, when it's on nice and tight to this spool, to this bead? So this is when these tails, they come into play, the tails of these histones. So remember what we said, they have these long N-terminal tails. So over here, some of them are, we can have, at these ends, we can have phosphorylation, acetylation, or methylation that can take place over here. And again, different enzymes, different proteins will come and interact with them under different circumstances, depending on, whatever job it's trying to get done, whatever the purpose is. Different proteins will come and interact with them at that point. So when you look over here, in terms of the ATP-dependent remodeling, there's many reasons that they kind of, and they're even giving an example here. So sometimes you can have where the nucleosome gets, in other words, the histones get evicted. So now you just have a run of DNA that's over here. Other times you can have some DNA sliding that takes place also. Or again, if you want to assemble, you want to put in nucleosome. It needs to be done through this process as well. If you want to unwrap it, okay, as for example, I told you before, perhaps you want to, you want some translation to take place. So again, that could be done through ATP-dependent chromatin remodeling as well. Well, what about maybe this histone's been damaged, you need to replace it. So at that point, again, you need to get rid of the bad one and put in a new one. So again, this is another point where another example of when this can take place. So in other times, you have a complete ejection of the dimer for whatever reason. Maybe perhaps you're having a new one coming in, an entirely new dimer. These are all several reasons and a few different methods as to how histone modification can take place. So thank you so much for watching this. I hope this made sense to you guys. If not, please feel free to email me and ask me any questions. Thank you again.